muscles, the muscles are going to generate force. They're going to contract to generate force and allow movement. And you recall that we have three different types of muscle. We have skeletal, we have smooth, and we have cardiac muscle. We're going to spend the bulk of our time on skeletal muscle, but we will also give an overview of the other two types. As we get started, let's do a little comparison between the three types of skeletal muscle. First of all, some histological differences and as well as functional. Here we see some photographs at the top and then some diagrams at the bottom. So we have skeletal, cardiac, and smooth. Skeletal muscles, muscle cells can be called muscle fibers because they're so long. It's kind of a confusing term, but this is a muscle cell or a muscle fiber here. Here's another one, and here's another one. So the skeletal muscle fibers or cells are very long. They're the longest of the three, and they're shaped like a cylinder. So like a paper towel roll or what have you. They have this cylindrical shape. They are multinucleated. They have lots of nuclei per cell. And the nuclei that you can see here, these dark structures, are at the edge of the skeletal muscle cells. Now sometimes you say, well that looks kind of more in the middle, but it's on the edge of the cylinder. So if you have a cylinder, they're on the outer edge of that cylinder. They're not in the middle for skeletal muscle. The arrangement of the proteins in skeletal muscle is quite uniform into structures called sarcomeres that we'll look at. And this uniform arrangement of the proteins gives this striated or banded pattern, dark and light banded pattern. So this is a striated as well. Skeletal muscle is voluntarily controlled. We can voluntarily control it. Skeletal muscle attaches to our skeleton, which is where it gets its name and helps us move voluntarily. And it also attaches to our eyes and can move the eyes. So that's skeletal muscle. And again, we're going to go into the greatest depth about how skeletal muscle works and functions. Cardiac muscle also has some sarcomeres, and so it's also striated, the organization. The striations are more difficult to see, but if we look closely, like where the arrow's pointing here, or here, I can see it on this monitor a little bit better than up there, but there are striations. There's pretty good there. You can see they're not as pronounced as the skeletal muscle, but it does have these light and dark bands, which are the striations. They do have intercalated discs, which are these lines here, 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 that not only holds the cardiac muscle cells together, but provides gap junctions so that ions, a wave of depolarization, can move from one cell to another. Cardiac muscle cells are shorter than skeletal, but longer than smooth. And they can have a branching pattern, as we see in several places here, branches of the fibers. And they have one, or possibly at the most two, nuclei per cell, and they're centrally located. Cardiac muscles are involuntary, and of course are only found in the heart. This is the heart muscle that contracts during systole. Quite a bit about. Well, finally we have the smooth muscle. Smooth muscle is named that because it doesn't have any striations. This is the shortest of all the muscle fibers. You can see in the diagram here. They're short and the ends are tapered. Tapered looks like this. sometimes we use the word spindle shaped. That just simply means they're tapered at their ends. And they have one nucleus per cell, and it's centrally located. Smooth muscle is found in the walls of our internal organs, what we call the viscera. So the stomach and the small intestine and the large intestine and the esophagus and the uterus and the urinary bladder and all of these kinds of, and of course they're going to move things through. For urine, they're going to move urine out of the urinary bladder. For the uterus, they're going to move the baby out during delivery. Another place that we can find smooth muscle is in the walls of the larger blood vessels. And they can cause vasoconstriction and vasodilation. And so the smooth muscle is there as well. So both cardiac and smooth are involuntary control. Skeletal is voluntary. And skeletal and cardiac are striated, smooth being non-striated. Well, let's turn our attention to skeletal muscle in more detail. This gives you some of the listing of the characteristics, some of which we've mentioned already. They're multinucleated. They contain a lot of mitochondria. Cardiac muscle contains a lot too. Whenever you need a lot of energy in a tissue type, you're going to have a lot of mitochondria, make a lot of ATP, and that's true for skeletal muscle and cardiac as well. They're going to have special invaginations of the membrane called transverse tubules, or T-tubules. We're going to talk about that quite a bit today. They have some proteins that are arranged in long fibers called myofibers. And these proteins are arranged in an orderly manner that we call sarcomeres. We want to learn about the sarcomeres. You've heard about them before, probably know the different types, bands and stuff, but we'll look at it again because to understand the function, it's very helpful if we know the sarcomere structure a bit. So we'll look at that today too and remind you of it if it's possibly reviewed. In muscle, we give different names for some of our structures of the cell. For instance, the plasma membrane we refer to as the sarcolemma. And the cytoplasm we refer to as the sarcoplasm. And the smooth endoplasmic reticulum we refer to as the sarcoplasmic reticulum. So these are just traditional historical names, but they're used all the time now still, so we sort of need to know what those terms mean, and that's what they mean. Myofibrils are proteins sort of bounded together in long fibers. We're going to see that we want to look at 4 main types of proteins in the skeletal muscle cell. The 2 that are most involved in contraction and force are myosin and actin. Myosin is thicker in diameter and myosin is thinner. So sometimes we say a thick filament is myosin and a thin myofilament is actin. They're arranged within the sarcomere in a particular order, and their interaction is going to generate the force of contraction. In addition to myosin and actin, we have 2 regulatory proteins that are important. And these are called tropomyosin and troponin. These are in the notes, so if you don't feel, you can write it down certainly, but if we will be covering them. Just to give you a review or a quick reminder, tropomyosin lies on top of actin and prevents the myosin cross bridge from binding to the actin, and troponin sits on top of the tropomyosin, and so they're preventing the interaction of myosin and actin. They're regulatory proteins. An increase in the calcium in the sarcoplasm will bind to troponin, cause a conformational shape, and slide that quite a bit today, but that was just a quick summary of it as we're getting deeper into it. This is our sarcomere. Here's our muscle fiber in the cell, and we have these actin and myosin running the length, and they're in a pattern that we call the sarcomere. The sarcomere goes from one Z line to another, and this structure in brown on our diagram represents myosin, the larger diameter protein, and this thinner in purple represents actin. If we blow the actin up a bit, we can see that this little blue ribbon here, that's tropomyosin, and the little pinkish in the diagram is troponin. Tropomyosin and troponin are going to block binding sites on the actin where these cross bridges of myosin would like to bind. This is an actual electron micrograph of a sarcomere in a skeletal muscle cell or fiber. And so one sarcomere, this is a Z line right here, as you can see, and here's another Z line. One sarcomere goes from Z line to Z line, and then we can name these other regions. These lighter regions that encompass two sarcomeres, such as here and here, these are called the I band, and they contain only actin, the thin myofilament. You can see it on our diagram down here. Here's the Z line, and here's the I band. Here's the other I band over on the opposite side, and the Z line. And then where it's this thicker part, this darker part from here to here, that's called the A band, and that's where you have both myosin and actin. And then right in the middle of a resting sarcomere, you have this H zone, which is only myosin, and the very, very middle you have the M line. So here's your M line that comes all the way up. We like to know the parts of the sarcomere again so it can help us relate to the function. We have different types of muscle contraction. On Wednesday, we'll go into this in a little greater detail, but you can have isotonic contraction where you move your muscles and you move your joints through a range of motion. And you can have isometric where you're contracting, but you're not moving the muscles through a range of motion. But in both cases, you're generating a contractile force. Not on Wednesday, but on Tuesday, tomorrow, we'll go over that in a little bit greater detail. So we're going to generate force by interacting myosin and actin. And so we look at our sarcomere here. What's going to happen is these myosin cross bridges are going to bind to actin, and they're going to pull up and slide actin in towards the center of the sarcomere and reduce the H zone and the I band, and that will generate a force. These cross bridges, they bind to the actin and slide, pull, and then they quickly disengage and bind. This is a very, very rapid process. It's just going, and they're all working together. Sometimes we use the analogy of oars in a boat. Rowing, and basically it requires ATP, and you may remember that ATP is required. It binds to the myosin cross bridge, and it phosphorylates the myosin cross bridge and transfers some energy from the ATP to the myosin cross bridge. But the binding of the myosin cross bridge to the actin requires that ATP to have already attached to the cross bridge. Now we're going to show you several videos. I think they're not very long, but I think the visual aspect here really helps me. hope it helps you too. Now I'm seeing if it's gonna actually open it. Well, maybe we won't be watching. Worked on my office computer. All right. Well. Unfortunate. We have several of these today. Give me an arrow to open it. No worries. So what happens is this cross bridge is going to bind to the actin and going to slide it towards the center of the sarcomere, and it generates a force, and it disengages and binds a little bit further down the length of the actin and causes this contractile force. Here's our actin. It has a binding site for myosin, and it can, the actin is twisted, like you can see in this diagram a bit. On top of the actin, in the resting state, we have tropomyosin, and it lies right on top between the actin and the myosin, cross bridges, and it's blocking the interaction of myosin and actin. Troponin sits on top of the tropomyosin, and it is also a regulatory protein. Essentially, as you probably recall from the past, troponin can bind calcium. When the intracellular calcium levels rise in the sarcoplasm, essentially, calcium binds to troponin, changing its shape, its conformation, and sliding tropomyosin out of the way. This allows the cross bridge to bind to actin. So calcium disinhibits this troponin tropomyosin complex and allows muscle contraction to start. Well, this one may work. Let's see.
Myofilaments are composed of strings of the proteins actin and myosin. An actin filament has two strands of actin molecules wrapped together. A myosin filament has many myosin proteins packed together, and each myosin protein has a globular head region that protrudes from the filament. Myofilaments can contract or shorten due to interactions between the myosin heads and the actin filaments. Contraction begins with the head of myosin molecules bound to actin on the actin filament. While still bound to actin, the myosin head flexes, pulling the actin filament along with it. This causes the actin filament to slide by the myosin filament. Then the myosin head releases from the actin and unflexes, a change that is powered by ATP. This frees the myosin head to bind with a different actin molecule farther up the actin filament. The entire process may then be repeated many times. The myosin, in effect, walks along the actin filament, moving the actin filament more each time.
Okay, and here it shows the actin with the tropomyosin and troponin. Here it's magnified. So the actin, the tropomyosin is sitting on top of these binding sites where the cross bridges of the myosin want to bind. So it's blocking those, and here's your troponin. Calcium coming in will bind here and slide this complex down so that the cross bridge can bind to these binding sites on the actin. The myosin cross bridge needs a binding site for ATP as well as the actin. So you have an actin binding site and an ATP binding site. Sometimes we call these cross bridges of the myosin heavy myromyosin, so if you've heard that term, it's the same thing. Now, when you're getting ready for this contractile event, you're going to have an increase in calcium in the sarcoplasm, and that's going to slide the troponin tropomyosin off. And you're going to have a cross bridge that is energized. The ATP will have attached to it, and it will have been hydrolyzed to ADP and inorganic phosphate, but the energy will have been transferred over to this cross bridge. And then that complex is going to bind, and using the energy that has been transferred over to the cross bridge, it will slide, it will bind to the actin and slide it towards the center of the sarcomere. To disengage, so it can re-engage, you have to have ATP binding to that cross bridge that has already done its work of sliding the actin, and so you bind another ATP on there, and that allows it to disengage. Again, you have a hydrolysis of the ATP, you transfer the energy to the cross bridge, and then it reattaches, and you keep that cycle going very rapidly. We all know that ATP is needed for energy, for muscle contraction, but we also probably are familiar that ATP is needed to disengage the cross bridge from the actin. And so after death, if a person has some muscles contracted, let's say they're sitting down or something like that, after death they're not generating ATP, and so they cannot disengage the cross bridges from the myosin. And so they remain in a stiffened position with those muscles still being partly contracted. And of course we call that rigor mortis, the rigor of death. Eventually those proteins degenerate and break down. But for a while there is this stiffness, and they can be difficult to move even to stretch out and things. So that's the rigor mortis that this little diagram here is referring to. This shows it in a little bit larger diagram. If the muscle is relaxed, we have a low cytosolic calcium. Tropomyosin is covering the binding sites. Troponin is sitting on top of the tropomyosin. And then we increase the cytosolic sarcoplasmic calcium, binds to the troponin. It slides the troponin tropomyosin out of the way, and this allows for the cross bridge that has been energized with the ATP to bind and initiate contraction.
During contraction of a muscle, calcium ions bind to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin heads attach to the exposed binding sites on the actin myofilaments to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to slide past the myosin myofilament. The ADP is released from the myosin head as it moves. The bond between actin and the myosin head is broken when an ATP molecule binds to the myosin head. The ATP is broken down to ADP and phosphate, releasing energy, which is stored in the myosin head and will be used later for movement. The head of the myosin molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
And that would be repeated as long as calcium and you had enough ATP to continue it. Eventually you get fatigue as you run out of ATP, for instance, other problems. Now, one of the things, what is going to induce the muscle contraction? Well, we're going to get action potentials down motor neurons, and then we're going to get action potentials along the muscle in the sarcolemma. And that muscle fiber action potential will cause the muscle contraction. There's a little bit of a latent period in between the stimulus and the contraction, and then we see the tension that is generated. We'll deal with this again more tomorrow. We'll go back more over the mechanism right now, but this is where we're heading tomorrow. So far, do you feel comfy, or anything you want to clarify or chat about? Or you're okay? You've probably heard some of this before, I suppose, so it's a good review. But well, let's talk about how we're going to increase the sarcoplasmic calcium, the level of calcium inside the cell. And to do that, we need to remind ourselves of the smooth endoplasmic reticulum, which in muscle cells we call the sarcoplasmic reticulum. This is a membrane-bound organelle, the sarcoplasmic reticulum. And in this membrane, it has calcium pumps, which, using ATP for energy, pump calcium out of the sarcoplasm into the sarcoplasmic reticulum. Now, it does this mainly at the ends of the sarcoplasmic reticulum, and these areas of the sarcoplasmic reticulum are called the lateral sacs. So here you see that term, the lateral sacs. So we are concentrating calcium in the lateral sacs of the sarcoplasmic reticulum. So it's waiting there. Here you see the ATP that's being used for that energy requiring process. Now, this pumping of calcium into the sarcoplasmic reticulum, the lateral sacs specifically, lowers the calcium concentration in the sarcoplasm. So how are we going to activate it? Well, we get a motor, action potentials in motor neurons, which come to the muscle, and then they're going to generate action potentials along the sarcolemma of the skeletal muscle. And they go along the length of the skeletal muscle, sarcolemma, but they go into the interior of that cylinder along invaginations of the sarcolemma called transverse tubules, or T-tubules for short. Here they are invaginating in. So as the action potential runs down the length of the sarcolemma, and then it runs into the interior down the T-tubules, when it reaches the lateral sacs of the sarcoplasmic reticulum, it opens some calcium gates. This is a rhyodine receptor, voltage controlled receptor. So when the action potential reaches there, it opens this calcium gate. Calcium diffuses down its concentration gradient into the sarcoplasm, and this raises the concentration of calcium in the sarcoplasm. Calcium binds to the troponin, changes its conformation, slides the tropomyosin away, the myosin cross bridge or head binds to the actin and initiates contraction using ATP for the energy source. Eventually, as the action potentials stop, the gates for calcium close, and there in the background, you keep pumping calcium back out of the sarcoplasm into the lateral sacs of the sarcoplasmic reticulum. And as you decrease the concentration of calcium in the sarcoplasm. Calcium disengages from troponin, and tropomyosin troponin slide back and inhibit the binding again, and the muscle contraction is over. So we need adequate amounts of calcium for muscle contraction as well.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the ends of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting in an increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long filamentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin myofilament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
So this is reminding us that the sarcoplasmic reticulum stores calcium, and it's actively transported in there, pumped in there. And the T-tubule carrying the action potential down, you're going to have a voltage-sensitive calcium gate or channel, dihydropyridine receptor. It's going to sense that voltage change and open the calcium gate or channel and allow those actions to take place. A motor unit, when we have a skeletal muscle, it can have thousands of muscle cells and muscle fibers. One motor neuron doesn't innervate all of those muscle cells in the skeletal muscle. Instead, one motor neuron innervates a subclass of the muscle cells in the muscle, and then you have another motor neuron that does some others, and that pattern is repeated. Now, the motor neuron and the muscle cells that it innervates is called a motor unit. Each of these motor units has a different threshold. Some can be activated at a lower threshold, some need a higher threshold in order to be activated. What this does is it gives us a gradation of muscle contraction. If we want to pick up a light load, a light object, we can activate only a few motor units, so a few of the muscle cells in that muscle. If we want to pick up something heavier, we would reach the higher threshold motor units, and we could activate a higher percentage of those skeletal muscle fibers until you've got all of the skeletal muscle fibers in a muscle contracting for the heaviest loads. Okay, that would be for one muscle. Then you could use other muscles, of course. You'll never use just one muscle. But the idea is within a single muscle, you can activate smaller or larger numbers of motor units depending on how much force you need to generate for that particular load. This shows you some motor units kind of color-coded. And so, again, these motor units will have different thresholds, and you can activate more and more of them as needed. Still okay with that? Okay. Well, what about the neuromuscular junction? This would be the somatic nervous system rather than the autonomic nervous system. And so you have a single motor neuron going from your central nervous system, either the spinal cord or the brain, to the muscle. When it reaches the muscle, you have a synapse. This is often called a neuromuscular junction. It has other names like motor end unit and some other things, but essentially it's a synapse or neuromuscular junction between the motor neuron and the skeletal muscle. Remember, we can have a number of motor neurons innervating a single muscle. They would innervate a different subset of cells within that skeletal muscle, and that would be our motor unit. When it reaches here, we notice that all of these somatic motor neurons are myelinated, and they're the largest diameter axons in the body. That means they carry the signal very quickly. And this is important because you need to move if there's danger. If your hand is on a hot stove, you need to move it quickly. If you're stepping on a tack, you need to move it quickly. So you need to have quick responsiveness, and this helps with that. The neuromuscular junction is always excitatory. It's always acetylcholine is the neurotransmitter in all of these neuromuscular junctions. And they bind to nicotinic cholinergic receptors. We talked about that in the nervous system, but we're kind of refreshing our memory of that.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium ion channels to open, increasing the calcium ion permeability of the presynaptic terminal cell membrane. Calcium ions enter the presynaptic terminal and cause vesicles to release their neurotransmitter acetylcholine from the synaptic vesicles into the presynaptic cleft. Diffusion of acetylcholine across the synaptic cleft and binding of acetylcholine to acetylcholine receptors on the postsynaptic muscle fiber membrane causes an increase in the permeability of ligand-gated sodium ion channels. The movement of sodium ions into the muscle cell results in depolarization of the postsynaptic membrane. Once threshold has been reached, a postsynaptic action potential is generated and is propagated over the muscle cell membrane. Acetylcholine is rapidly broken down to acetic acid and choline in the synaptic cleft by the enzyme acetylcholine esterase. The choline is reabsorbed by the presynaptic terminal and combined with acetic acid to form more acetylcholine, which enters the synaptic vesicles.
This shows you what was being described there. Here comes the action potential. This is myelin here. It's coming down. When it reaches the terminal of the axon here at the neuromuscular junction, the action potential's arrival causes the opening of voltage-regulated calcium gates in the axon terminus. Calcium diffuses in. That initiates the movement of the vesicles containing acetylcholine to the membrane. By exocytosis, you release acetylcholine. It diffuses across that small space, the synapse. It binds to nicotinic acetylcholinergic receptors, and that then causes the sodium to come in, and you initiate action potentials in the membrane, the sarcolemma now, of the muscle, and it goes in both directions. In the nerve, it only goes unidirectionally because the gates proximal to it, the signal are in absolute refractory period, but you go sort of in the middle of the skeletal muscle, and so it goes in both directions, and then it goes internally with the transverse tubules and initiates the release of calcium from the sarcoplasmic reticulum. Of course, we need to have negative feedback, so the acetylcholinesterase breaks down the acetylcholine into the component acetate and choline. Choline gets taken up, and the acetate goes in the blood and eventually gets back here over time. And so that deactivates it. Now we have this sequence, this is from your book, these next 2 pages, they list the entire sequence going from the motor action, neuron action potential, all the way through relaxation, contraction, and back to relaxation again. So this is the overview. How can we disrupt that signal? Well, curare is a poison that can bind to the nicotinic cholinergic receptors, but not open sodium gates, and not be broken down by acetylcholinesterase, so it essentially blocks the muscle contraction at that step, and that causes paralysis. The muscles can't contract, and because we need skeletal muscles to breathe, pull that thoracic cage up, this causes asphyxiation. Oftentimes in South America, they would dip the little arrows in curare, either using a blowtorch or other blow gun, I should say, or arrow, bow and arrow kind of thing, and they would kill the animal that way. They would hunt them, and so that's how they would easily do their hunting. So that's curare. There are certain nerve gases like saffron, not saffron, sarin, sorry. Saffron the spices. we won't say that. Saffron will kill you. No, sarin gas. That can bind, and this can also result in skeletal muscle paralysis. In this case, they inhibit acetylcholinesterase, and so they leave the acetylcholine on there too long, so it's contracting, but it's sustained contraction. So you have to have both a breathing in, but you have to relax it so you can breathe out. So this is also a problem in that case. All these are nicotinic cholinergic receptors at the skeletal muscle, but just as an aside, these nerve gases can also build up at the muscarinic synapses, such as at the heart, and inhibit the heart, and that can cause the heart to fail. You can try to give atropine to counteract that effect. We also are aware that there is a bacterial caused disease caused by Clostridium botulinum, and it causes botulism. And this affects the release of the acetylcholine. It disrupts the so-called snare complex that is required for the vesicle containing the acetylcholine in the motor neuron to bind and fuse to the membrane and release the acetylcholine. If you get this form of botulism, it can cause death because of, again, not allowing the release of the acetylcholine. However, as we've talked chapter, you can use small amounts of botulinum toxin injections to smooth out the skin, smooth out the face, Botox, and if there's excessive blinking, you can inhibit that. You can also use it if there's excessive sweating. Somebody has a disorder where they're just sweating excessively. So you can use it clinically for positive if you, of course, keep it in lower doses and you don't inject it systemically. You can do that. Treatment of migraine, I'd sort of forgotten about that one, but acetylcholine is somewhat implicated in migraine headaches, so that's another thing that it could be used for. All right. Do we still feel comfy? Think we could write all that out, how this works, if we had to?
I think I will.
Yeah. Have to review it, right? Fresh in your mind, but, that's how it works. So tomorrow, we're going to go over, just as a quick preview, we're gonna go over the forces of contraction for muscle. We're gonna look at isotonic and isometric contractions and compare them. And we're going to look at tetanus, diffuse tetanus. Saw some of that in the muscle video if you've watched them already. And then we're gonna look at the energetics about it, and fatigue, and all of that. Then we're gonna look at different types of skeletal muscle fibers genetically, and the characteristics there. And then we'll look at more skeletal muscle disorders, and then we'll look at smooth muscle and cardiac muscle. That'll be the next 2 days for all that. But that's just to show you kind of where we're heading over the next 2 days. And then, of course, the test on Friday will include this muscle chapter as well as the final chapter. So we've got muscle, respiratory, cardiovascular. Anything else? This test? Oh, sensory. Thank you. Yeah, sensory. That was not that long ago, was it? Yeah, sensory. Just bang, sensory. So there's 4 pretty big chapters on this, 4 big subjects. So the format of the test will be pretty similar to the last one. It'll be about 100 points. There'll be multiple choice, multiple choice, same style, and some short answer and some essay. There will be 1 case study on there. And so, yeah, that's the things to prepare for Friday. It's a lot, isn't it, to try to remember all of that, but that's coming up on Thursday. I don't know why I keep saying Friday. Thursday. Remember, we can here at 2 o'clock for the test on Thursday. During lap time, and we have 2 hours to do the test. Okay, wonderful. Great to see you again.